Hello, hi, a very happy good evening. So this is Krishnaveni and we've been talking about the story of life on earth, and that's biology. Yes, welcome to my session. So today we're going to discuss the previous year questions of class 12 topics. So we have been doing the year 2020. So today we will finish up 2020 and begin with 2019. So let's see how the pattern is. So there is not much difference between 2019 and 2020 paper. So almost it has the same set of questions. So from 12th standard, you're having only 43 questions in both the years. So let's see how the questions differ. Yes, Dharani, a very happy good evening. Thank you so much for joining my lecture. Yes, Anthony, hello, hi, a very happy good evening. I'm good, thank you. How are you? So hi, Vaishali, a very happy good evening. Thank you so much, all of you, for joining my session. So please do like, share and subscribe. Okay, so without any further delay, I'm moving forward. So today we are having a rapid fire mentee in the topic Mendelian genetics. So the entire topic, so Mendelian genetics is nothing but your principles of inheritance and variation. So you will have 20 questions. So some questions will have only 10 seconds. Some questions I have given 60 seconds, that is 1 minute. So it depends on the difficulty level of question, and only once you will get the leaderboard. So it will be very quick. Okay, it's rapid fire. So it's almost, you'll just play for hardly 10-15 minutes and then we'll be done. So please join me for this rapid fire mentee. So this is at 6:30 p.m. this evening. So this is at 6:30 p.m. this evening. So please join me for this rapid fire mentee. Yes, so please do share the link with your friends. Let your friends know, yes, we are having a rapid fire mentee. So all the theory questions, you will get only 12 seconds or 10 seconds. But the questions which involves crosses, yes, I have given ample time. So you should be able to solve within that, provided having taught you so much of questions, having solved so many questions in class, it should be easy. Okay. Ma'am, to pursue career in veterinary science is nice, absolutely nice, because veterinary doctors are also in demand. Veterinary doctors are also in need. So only when you have a pet dog or a pet animal, you will know what is the demand for a veterinary doctor, because before that even I didn't know so that veterinary doctors are so important. I thought hardly 1 or 2 is there, but to know each and every animal has to get vaccinated. So always, so here also when I take a street dog to a doctor, so I look for veterinary doctors. So quota is new to me, but still here also I found veterinary doctors. So it is a very good field. So veterinary doctors are needed throughout. So even if you go to forest department, there people look for veterinary doctors. So you have a career in the government genre as well. Yes, of course, veterinary science has a very good scope ahead. Okay. Yes, hello, hi Shimpi, a very happy good evening. I'm good, thank you. How are you? So thank you, Shimpi, for joining my lecture. So please do like, share and subscribe. Hi Keshav, yes, hello, thank you for joining my session. So please everyone join my mentee as well this evening. So it is at 6:30 p.m. Okay, so look who is here after a very long time. Yes, Priyanka, a very happy good evening. I hope you're doing good. So thank you for joining my lecture today. So please do like, share and subscribe. Okay, so we were in the year 2020, so we were discussing these questions. So let's finish off 2020 first. So this is the order in which we are going to discuss. Yes, so we have already discussed genetics and evolution. So we have also discussed biotechnology. So we have discussed ecology. So only these two are pending in the year 2020. So now let's discuss reproduction, biology and human welfare, and then move forward with the year 2019. Yes, okay. Yes, fine. So this is the chapter wise weightage that I have given you. Yes, if you can see, biotechnology 1 has the maximum weightage, and then you have your human reproduction. So 3 questions here and 2 questions here. So and then of course you have molecular basis, which has the high weightage. Okay. So let's move forward. So we were here, we are supposed to discuss reproduction. Yes, Munna, hello, hi. So how sweet of you, you were on time today. So yes, I'm good, thank you. How are you? So come on guys, please do like, share and subscribe the session. Okay, so let's go to question number 1 of reproduction. Okay, so I guess this question we have discussed. So this is option 2. Yes. No, this is not option 2. This is option 3. A and D was the answer. This we have discussed. So this also, I guess Shimpi has given me the right answer. This is insects or wind. So that is the pollination. Okay, so question number 3, I guess I stopped here. So the body of the ovule is fused within the funicle at which position? Micropyle, nucellus, chalacal, hilum. So when the body of the ovule is fused with the funicle, what is that portion known as? Yes, Keshav, absolutely right. So it is known as hilum. So this is option D. I guess we stopped with this question, right? Okay. So here we move forward with question number 4. Yes, give me the answer. Which of the following hormone levels will cause the release of ovum from the Graafian follicle? High concentration of progesterone, low concentration of LH, low concentration of FSH, high concentration of estrogen. Yes, what is the answer here? Yes, quick. High concentration of estrogen. Yes, question 4. What is the answer, guys? So guys, please do like, share and subscribe. So which of the following hormone levels will cause the release of ovum from the Graafian follicle? Yes. Is it? So it is not low concentration. So some high levels will cause the release. So it is not B and C. It should be between A and D. So Priyanka says D. So let's look at the right answer. Yes, it is high concentration of estrogen as well as high concentration of your LH plus your FSH. So this induces the rupture of Graafian follicles, thus releasing your ovum. Yes, Munna, absolutely right. Very good. So moving forward to question number 5. Meiotic division of secondary oocyte is completed at the time of copulation, after zygote formation, at the time of fusion of a sperm with an ovum, and prior to ovulation. So the same question has been repeated in the year 2019 as well. So it is very important. Meiotic division of the secondary oocyte is completed at the time of copulation, after zygote formation, at the time of fusion of a sperm with an ovum, prior to ovulation. Yes, what is the answer? Yes. So Priyanka says option C. Yes, others. So Shimpi, what is the answer? Yes, so at the time of fusion of a sperm with an ovum, the second meiotic division is completed. Yes, absolutely right. So question number 6. Match the following columns and select the correct option. Okay. So this is also from human reproduction. So placenta, zona pellucidia, bulbourethral gland, Leydig cells. Yes, come on, quickly mark the options and tell me what is the right answer. Yes, come on, quick. So placenta secretes HCG. Okay, so this should be A. So zona pellucidia is the layer of ovum. Okay, so bulbourethral glands. So bulbourethral glands are present in male. So this is C, and Leydig cells secrete androgens. So A is 2. Okay, so C is the right answer. Okay, yes, Priyanka is right. So Keshav is right. So I have Loga Satish. So hello, hi, welcome. Thank you for joining my lecture. So please do like, share and subscribe. Yes, all of you are right. So option 3 is the right answer. So does anyone have any doubts here? So should I explain? Yes. So placenta secretes HCG, so human chorionic gonadotropin. So zona pellucidia is the layer of your ovum. Bulbourethral gland is a gland, accessory gland present in your male, and Leydig cells secrete androgens. Okay? Yes. So question 7. In which of the following technique the embryos are transferred to assist those females who cannot conceive? Yes, which of the technique? So I guess you know what is GIFT. So this is gamete intrafallopian transfer. So this is your GIFT. Then you have ZIFT. This is zygote intrafallopian transfer. Then you have ICSI. This is intracytoplasmic sperm injection. Right, so IUD is intrauterine transfer. Yes, which is the one? Which is the answer? Who cannot conceive? So what do you do? Yes, Yes, option D is the right answer. So you do zygote, so you do in vitro fertilization and then you transfer it back to the female, right? So they are not able to enhance the fertilization process, so you do that artificially. So zygote intrafallopian transfer and intrauterine transfer, that is IUT. Okay, good. So question number 8. So select the option including all sexually transmitted diseases: gonorrhea, malaria, genital herpes, AIDS, malaria, filaria, cancer, AIDS, syphilis, gonorrhea, syphilis, genital herpes. I'm sorry, I gave the answer. It's pretty easy, right? So malaria is not genetically transmitted. So malaria and filaria is also not genetically transmitted. Cancer is not genetically, so it's sexually transmitted. So only this is the answer. So this is a very easy question for all those who have attended 2020 paper. So this should have been an easy case. At least even if you have made a mistake, now it should be easy. Right? So moving forward to question now. Okay, so we are done with the 8 questions in reproduction. Now we come to the next unit, that is biology in human welfare. So here we go. Come on, quick, give me the answer 1. Match the following diseases with the causative organism and select the correct option. Yes, match it and give me the right answer. Yes, take your time. So once I will have a Menti where I will only have match the following, okay? Yes, of course. So your NTA makes the question paper tough, but actually the NEET papers are pretty easy. Yes, you're right. Okay, so typhoid is caused by salmonella. So pneumonia is caused by Haemophilus influenzae. So this is B. So filariasis is your Ucheria. Malaria is Plasmodium. So this is such an easy match. So what is the right answer? So A is 3. Okay, so option 1 is the right answer. Yes, very good. Okay, I hope everyone got it and there are no doubts. Okay. So question 2, the infectious stage of Plasmodium that enters the human body is sporocyte, female gametocyte, male gametocyte and tropozoites. Yes? Yes? Sporozoites. Yes, others? Yes, the right answer is sporozoites. That is your infectious stage for all those who do not know the life cycle of Plasmodium. So Plasmodium is a type of protozoan that causes your malarial disease. So how does this disease happen? So protozoans mostly live as parasites. Ma'am, which one? High incubation period in malarial causative agents. High incubation period means, so you have your severe malarial fever. So that has the high incubation period. Okay, here a quick recap of the Plasmodium cycle of malaria. So Plasmodium is a protozoan. So it causes malaria. So protozoans are mostly parasites. Okay, so yes, Plasmodium falciparum is one strain that causes malignant malaria. So this malignant malaria can be very, very fatal. Plasmodium vivax is benign. It has mild symptoms. So Plasmodium falciparum lasts longer. Okay, yes, so they are basically parasites. So they need 2 hosts. One is your mosquito and the other one is your humans. So we all know mosquitoes feed on human blood. So mostly these parasites are present inside the mosquito's salivary glands. So I am talking about the parasite. The parasite is present inside the mosquito's salivary glands. Okay, they are present in an infectious form. So in the life cycle of this Plasmodium, there is one particular infectious stage that is known as sporozoites. Okay, so they are present inside the they are present inside the mosquito. So here the mosquito is female Anopheles mosquito. Female Anopheles mosquito. Okay, so this mosquito bites a human. So the sporozoites enter inside the human. They quickly replicate inside the liver of the man and they become mature. When they are mature, they are known as scyzons. Okay, they are known as scyzons. Fine. So these scyzons then enter your blood circulation, especially your RBC. okay, and they multiply into a higher number. So here they mature, and in RBC they multiply into higher number. So in multiplication, I mean cell division is happening. A mother cell is moving or dividing into daughter cells, right? So here inside the RBC, they produce daughter cells known as merozoites. So finally, when the RBC can no longer manage it, the RB ruptures and these merozoites are released out. So now this RBC is rupturing. So along with this, a toxin is released that is known as hemozoin. So that toxin causes your malarial fever. Okay, so now these merozoites are ready for your sexual reproduction. So they enter your gametophyte stage. So they form the micro male gamete and the macro female gamete. So once again, when the female infects or feeds on this human, the gametes are passed on into the mosquito itself, and your sexual reproduction or fertilization happens inside the mid-gut of your mosquito. So once again it becomes very big, it becomes a full adult, and it becomes a sporocyte and gets stored into the salivary gland of your mosquito. So this is the life cycle of your plasmodium. Yes ma'am, a very happy good evening. Thank you for joining my lecture. So please do like, share and subscribe. How to read plant physiology easily. So that is with a lot of interest. So that would be my genuine answer. So I cannot force someone to read something, right? As long as you do not have interest, then it will not make the entire story interesting. Even if you ask me or push me into the movie of Marvel, I will not find any interest, because that is how it is. Okay, so it's like that. So reproduction in liver. Okay. Talk to plant. Okay, yes, so Shimpi remembers it. So I hope you have understood the stage of your plasmodium. So very soon I'll be teaching this topic, but I just gave a rough outline here. Yes, question number 3. So identify the wrong statement with reference to immunity. So I want the wrong statement identified. When ready-made antibodies are directly given, it is called as passive immunity. Active immunity is quick and it gives full response. Fetus receives some antibody from the mother. It is an example for passive immunity. When exposed to antigen, antibodies are produced in the host body. It is called as active immunity. So which is the wrong statement here? Yes, which is this wrong statement? Yes, Satyendra, hello, hi, a very happy good evening. So thank you for joining my lecture today. So please do like, share and subscribe. Yes, so active immunity is not quick. Active immunity is slow or gradual, but it gives full response. This is right. So thus this entire statement is wrong. Thus the right answer is option 2. Yes, good. So question 4. So actually this comes in human physiology, but I somehow skipped it and took it under your human health and disease. But still, as long as you can answer, it does not matter. Yes, match the following columns and select the correct answer. Yes, now give me the right answer. Passive is quick and it's short-lived. Yes, you're right, Keshav. So eosinophils, basophils, neutrophils, lymphocytes. Yes. Any idea? So Antonia B says 4. Okay. So basophils will release your histamine and destructive enzymes. Okay. So before that, these are all components of your leukocytes. So leukocytes are your WBCs. So WBCs is broadly divided into your granulocytes and your agranulocytes. So granulocytes are BEN. So this is basophil, eosophil and neutrophil. So basophil because it stains with a basic dye, eosophil because it stains with an acidic dye, and this does not stain with any dye. That's why it's called as a neutrophil. Agranulocyte is lymphocytes and your monocyte. So lymphocyte induces immune response. So this is D. So here look into the options. So 1 you know properly because lymphocytes are your B lymphocytes and your T lymphocytes. They trigger your immune response, right? So for lymphocytes, it's immune response that is fixed. So you go with one option. This is right. Yes. So this is the right answer. Now neutrophils, so neutrophils come under this category. Okay, so neutrophils are here. So neutrophils, they perform the function of phagocytosis. They do phagocytosis means they kill some cells which carry your antigen. So this is C. So now if you have a confusion between basophil and eosinophil, at least you know 2 options, then look into the remaining 3. So eosinophils release histamine, okay, so that is during allergic reaction. So allergic reaction is IgE, so remember E. So this is 3, so sorry, this is A and this is B. So the right answer here is option 4. Yes, the right answer here is option 4, okay. Phagocytosis means engulf of pathogen, okay. That's okay, Antonia B, as long as you're able to rectify your mistake, I'm fine. Okay, phagocytosis. So phago in a literal term is eating or engulfing. So cytosis means cell. Cyto is cell. So there will be some particular cells which carry antigen without their knowledge, okay, but the function of your T cells or some of your lymphocytes is to track these cells which carry a surface antigen, and these neutrophils or your phagocytic cell captures those cells and destroys them. So that is known as phagocytosis, okay. Yes, IgE is for allergy and E stands for epsilon. Yes, you're absolutely right. Okay, so Sai Ram, hello, hi. So thank you for joining my lectures today. So please do like, share and subscribe. Ma'am, when you start NEET 2022 batch, okay, so you can join whenever you want, because I have 1 round completed a syllabus. I am revising, I am asking questions, so I am discussing questions also. So gradually start joining. So whenever I am starting a new topic, so please schedule yourself so that you can join me, so you can cover up that topic. So you don't have to wait for 2020, the batch will start in October, so you don't have to wait till then. So I'm not saying it will start in October, I'm just giving you a tentative date, because as of now we don't know when your 2021 exam is. So whenever I'm starting a new chapter, so please keep in touch so that you can finish that chapter, okay. Yes, fine. So question 5, by which method was a new breed Hisserdale of sheep formed using Bikaner apes and Merino rams produced? So mutational breeding, crossbreeding, inbreeding, outbreeding. Yes, what is the answer here? So crossbreeding, okay. So in animal breeding, so this question is from strategies for enhanced food production. So in this, so we spoke about animal breeding. So animal breeding is of 2 types. So animal breeding, so one is inbreeding, the other is outbreeding. So outbreeding means, so inbreeding is when you mate with the same type of breed for many generations, that is inbreeding. So outbreeding is of 3 types. One is outcross, the other one is crossbreed, the third one is interspecific or interspecies hybridization. So outcross is you breed between the same breeds, that is you mate between the same breeds of the same species, but they do not have a common ancestor. So imagine like arranged marriage. So arranged marriage is between a human and a human, right? So you do not marry a donkey. So between human and human, but the girl and the boy have different ancestors. So remember, this is like an arranged marriage, okay? So crossbreed is you take 2 different breeds of a particular species and then marry them. So let's take golden retriever and Labrador, and if you mate them, that is a crossbreed. So Hisserdale is an example of crossbreed because you're choosing 2 different breeds of the sheep and then doing, okay. Then interspecies is when you take 2 different species but yet they are related. So you take a donkey and a horse and you mate and you produce a mule, but you do not mate a donkey and a fish, right? So they are still closely related but 2 different species. So that is your interspecies hybridization, okay. Yes, you're right. So inbreeding increases pure characters, but it also leads to inbreeding depression. So outcross overcomes the inbreeding depression. Ma'am, in biology, which unit we have to start first if we do not have any knowledge in basic? So please start from class 11. So if possible, go in order of the chapters that is given, because the chapters are designed in such a way that it has a cohesive link with each of them. So please go in order if you do not have any basic. But yes, if you have some basic, then start with human physiology. So that would be my genuine suggestion. Okay? Yes? Anyways, thank you Satya for joining my lecture today. So please do like, share and subscribe. Yes sir, a very happy good evening. So thank you for joining my lecture. So please do like, share and subscribe. Yes, question number 6. So here we go. Which of the following is put into anaerobic sludge digester for further sewage treatment? Floating debris, effluents of primary treatment, activated sludge, primary sludge. Which of the following is put into anaerobic sludge digester for sewage treatment? Floating debris, effluents of primary treatment, activated sludge, primary sludge. So anaerobic sludge digested. So last one. Effluents of primary treatment? Yes. How it will be effluents of primary treatment? So this is talking about your sewage treatment, and this question is from microbes and human welfare. So what is the first tank? So you call it as your primary tank, right? So here it is also known as your mechanical treatment. Why? Because you pick out the floating debris by sedimentation and filtration, right? So if you do sedimentation and filtration here, so if this is my tank, yes, so if I let it to sediment, so there will be some solid particles which settles at the bottom. So this is known as my sludge, and this is known as my effluent. So since this is primary tank, this is primary effluent, and this is primary sludge. Okay, so now I take this primary effluent and put it inside my second tank. My secondary tank or the secondary treatment is also known as biological treatment, because here I am going to introduce microorganisms. What type of microorganisms? Aerobic microorganisms, because they can use oxygen from the organic matter present in my sewage water. Okay. So now after some time, here also I have some settlement. After this bacteria has grown in abundance, so they settle down, right? And then I have some fluid. So this is my secondary effluent, and this is my activated sludge. This is not called as secondary sludge, but this is called as activated sludge because so this has some microorganisms. So now I take this activated sludge and put it in the third tank, that is my anaerobic digester. So this aerobic bacteria are digested by anaerobes, which in turn gives me gases like methane, carbon dioxide, etc. So here the right answer is activated sludge. Okay. Yes. So question number 7. So match the following and select the correct option. So this is also from your microbes. Yes, give me the right answer now, quick. So one day I will bring a mentee where you will have only match the following. Okay. So I will do class 11 match the following or unit wise match the following mentee, so that will be easy. So now we are having guess the chapter. So this week we do not have any guess the chapter series, but next week you might have guess the chapter mentee series, because I have not covered all the topics, and then I will start match the following also has mentee. So Clostridium butyricum, so butylicum, so this produces my butyric acid. So Aspergillus niger is acetic acid. So Trichodoma polysporum, so it produces what? Yes. So yes, everyone has given me the right answer. Okay. So Clostridium butylicum, butyric acid. Okay. And Trichodoma polysporum, cyclosporin A. So C, monascus, blood cholesterol lowering agent, that is statins. So this is C and D. So this is the right answer. Yes, you're right guys. Perfect. So now we are done with the year 2020. So now we are discussing the year 2019. So questions from 2019. So I am discussing the previous year because I am done with the teaching the syllabus. So I have taught the entire class 11, class 12, botany and zoology in YouTube. So one round is over. Now I'm going with the second round in the name of syllabus wrap-up series. So that is why I'm discussing previous year question. For all those who are new and who are just beginning your NEET preparation, so please don't panic. So this is just a introduction or a recap to you, or you can say a flashback. Okay, so don't get panicked. So here 2019, so I'm sorry, this is 2019 analysis. So 2019 analysis, if you see genetics and evolution, you have 14 questions, the same number, but reproduction has a bit higher question, and these three have the same. So in case of your 2020, biotechnology had 9, ecology had 8, and this had 11, and this had 7. Okay, so this was the same, 7, 7. So this is 2020 analysis, this is 2019. So here they have paid a lot of attention to genetics and evolution. So Shimpi, don't worry. So Shimpi, you have not learned all the chapters. You have just joined me with the syllabus wrap-up series, so don't worry. Okay, so this is the analysis. So if you see, maximum is concentrated by genetics and evolution. So today in 2019 paper, we will discuss only genetics and evolution and reproduction. Yes? Okay, so chalo, let's go forward. So this is the chapter wise questions, number of questions. If you see, maximum question comes from principles of inheritance and variation. So that is my Mendelian genetics, and this is your molecular basis of inheritance. So this backs the maximum weightage. So human reproduction has only 2 questions and reproductive health has 3 questions here, and the rest, yes. So ecology, we have 4 chapters, right? So all the 4 chapters combined, we have only 7 questions. She is 12th now, so only like this, if you practice more, you will definitely do. So Shimpi is in class 11th, not in 12th. Yes, so Shimpi has just crossed 10th. She was Asad's student, and now she's come to 11th. Hannah, Shimpi? Yes? Okay, fine. So shall we start with genetics and evolution? So here we move forward. So question number 1, so you have to give me answers quickly. So what is the genetic disorder in which an individual has an overall masculine development, gynecomastia and is sterile? Edwards syndrome, Down syndrome, Turner syndrome, Klinefelter syndrome. Yes, come on quick. Yes. Klinefelter syndrome, yes, absolutely right, Keshav. Okay, so here an individual has an overall masculine development. So in case of Klinefelter syndrome, this is the genotype. So that is why the individual has overall masculine development. But since there are 2 X, so the part of female character also plays a role. It is not 47 plus XY. So it's totally 47 with 3 sets of sex chromosome. Okay, Keshav, it is not 47 plus 3, then it will account to 50, right? So that is wrong. So it is 47, in the bracket it is XXY. Okay, yes. So gynecomastia is the development of breast in male. So of course Klinefelter syndrome affects male and they are sterile. So basically this talks about your transgenders. So yes, this is option 4, that is the right answer. So question number 2, yes, my cup of cake. So in Antiherenum, that is snapdragon, a flower red was crossed with a white flower in F1 generation. So pink flowers were obtained. When the pink flowers were selfed, the F2 generation showed white, red and pink. Choose the incorrect statement from the following. So this is basically incomplete dominance, right? So ratio of F2 is 1/4, 2/4, 1/4. So they're just talking about the ratio. So law of segregation does not apply in this experiment. This experiment does not follow the principle of dominance. Pink color if in F2 generation is due to incomplete dominance. So I need the incorrect statement. So the F2, okay, so it is like this. So red color, white color. So this is red flower, this is white flower. For all those who do not know how to solve, so here the gamete is R, here it is R. So this is my F1 generation, this is pink in color. So I cross it. So I get 4 offsprings like this. So I have 2 pink. So I have 1 white, 2 pink and I have 1 red color. So this is my ratio. Yes, what is the answer? Law of segregation does not apply in this experiment. So that is the wrong statement, because yes, it applies. So this segregates independently of this, right? Yes, absolutely right. So question number 3, select the incorrect statement once again. So I want the incorrect statement. In domesticated fowls, sex of the progeny depend on the type of sperm rather than egg. Human males have one of their sex chromosome much shorter than the other. Male fruit fly is heterogametic. In male grasshoppers, 50% of the sperms have no sex chromosome. Yes. So I want the incorrect statement once again. So please make sure if they are asking the correct statement or the incorrect statement. That's okay, because even sometimes I don't read it properly. So that comes habitually. So we are so trained to look at only the right statements. That is the problem. Okay. So in domesticated fowls, the sex of the progeny depends upon the type of sperm rather than an egg. So domesticated fowls. So fowls include birds from whom we obtain egg and meat. So they are called as fowls. So in birds, it is ZW and ZZ type. So here the sex is determined by the female, right? So this is the wrong statement. Thus, according to the question, this is the right answer. In male grasshoppers, 50% of the sperms have no X chromosome. That is right, because that is a part in your grasshopper study. So don't have to confuse. Okay? Yes? Okay. So question number 4. So the frequency of recombination between gene pairs on the same chromosome as the measure of distance between genes was explained by. The frequency of recombination between gene pairs. So I have two genes, and what is the frequency of recombination between the two genes on the same chromosome as a measure of the distance between gene was explained by. So this talks about linkage and recombination. Yes, you're right, Dharani. So it means like your hen, duck, etc. Yes, who gave this? T.H. Morgan. Okay, so this I have not taught you. So the frequency of recombination. So T.H. Morgan actually found out linkage. Right? Yes. So this linkage was found by T.H. Morgan. But as a measure of distance between the genes and the frequency of recombination, you had another scientist also, that is Alfred. Yes, he was T.H. Morgan's student. So Morgan just gave an highlight and his student further developed on. His student was Alfred's two event. Okay? Yes, gene mapping. So question number 5. What map unit is adopted in the construction of genetic map? So I have solved this question recently in my lecture. Yes, give me the right answer. Yes, give me the right answer. A unit of distance between genes on chromosomes representing 1% crossover. A unit of distance between genes on chromosome representing 50% crossover. A unit of distance between two expressed genes representing 10% crossover or 100% crossover. I have discussed this recently. Yes, it is 1% crossover. Yes, absolutely right. So in the question that I discussed, A and B were interchanged, but it is 1%. No, it is 1% only. Yes, absolutely right. So question number 6, express sequence tag. So from here you have your molecular basis of inheritance beginning. So express sequence tag refers to DNA polymorphism, normal DNA sequences, genes expressed as RNA or polypeptide expression. So express sequence tag comes in your human genome project under the topic human genome project. So you tag the genes. So you have to sequence your whole genome, right? Yes. So this whole genome, you first segregate them as to which gene codes for protein, which gene codes for RNA. The ones which code for RNA are sequenced or labeled as express sequence tag. Yes, so Monday we are starting molecular basis of inheritance at 4 p.m. and at 6 p.m. we are discussing some important questions from the cell cycle and cell division. Alright, yes? Yes. So question number 7. Under which of the following conditions will there be no change in the reading frame of the following mRNA? So it is from 5' to 3'. Insertion of A and G at the 4th and the 5th positions respectively. Deletion of GGU from 7th, 8th, 9th position. Insertion of G at the 5th position, deletion of G from the 5th position. So a mRNA is read in a triplet fashion. So wherever I add and delete, this reading frame will be disturbed. But if I delete GGU directly, then it will not disturb. This will be one protein, this will be one protein, this will be one protein, this will be one protein. So if I delete this, there will be no change in the reading frame. Yes, Shimpi, absolutely right. So this is option 2. Okay, everyone clear? So question 8. So purines found in both DNA and RNA are, there can be no easy question than this in a national level exam. Guanine, cytosine, cytosine, thymine, adenine, thymine, adenine and guanine. So you have two types of nitrogenous bases in your nucleic acid. One is your purines, the other one is your pyrimidines. So purines are adenine and guanine, and this is cytosine, thymine in your DNA, and in case of your RNA it is cytosine and uracil. So here what is the answer? Yes, adenine and guanine. Yes, absolutely right. So question number 9, Yes, match the following genes of the lac operon with their respective products or produces. So lac operon is a type of a regulation of gene expression. Yes. Yes, that's a good trick. Okay, Priyanka. So as long as you remember it, I'm fine. So I gene is what? Z gene codes for beta-galactosidase, A gene codes for trans-acetylase. So this for permease, Y gene, and I gene produces your repressor protein. So that's the right answer is option A. Yes guys, absolutely right. So if you have not understood lac operon, so please join me on Monday session at 4 p.m. so that I'm starting the new chapter. So question 10, which of the following features of the genetic code does allow bacteria to produce human insulin by recombinant DNA technology? Genetic code is nearly universal, genetic code is specific, genetic code is non-ambiguous, and genetic code is redundant. Yes, which one? Which of the following features of the genetic code does allow bacteria to produce human insulin by recombinant DNA technology? Yes, quick. Yes, genetic code is universal. It codes, so U codes for phenylalanine in all living organisms. So C codes for proline in all living organisms. So this universal property of the genetic code enables us to easily produce human insulin by recombinant DNA technology. Match the hominoids with the correct brain size. So this is from the evolution topic. Homo habilis, Homo neanderthalis, Homo erectus and Homo sapiens. So this time when I teach evolution after your molecular basis of inheritance, so when I come to evolution, actually this time in evolution I have made a tabular column for these people. So they also have nicknames also, so I will give you that tabular column. So please remind me if I forget. Okay? Yes, what is the right answer? So this might be a bit confusing and tricky. Yes, but any guesses? So if you know who came first, then you know the smallest size belongs to them. So here the maximum size is 1400 cc. Cc is cubic centimeter. Is cubic centimeter. Okay, Shimpi? So Homo sapiens are the highest or the complex species. So they have 1400 cc. So looking at the option, it is easy to find. So though even if you don't remember this, it is still easily done. Because Homo sapiens are the last ones, right? So we are Homo sapiens. We do not have a much evolved being. So they have the highest brain size, that is 1400 cc. So only one option is matching. Oh no, we are wrong. So who has 1400 cc? Okay, Homo habilis. Okay, so I thought I was going wrong. Okay, so the trick is Homo sapiens are So if you compare the brain size of monkeys and of humans, there is a small twist in the story of evolution. So monkey has a huge brain size, but our brain size is compressed. But whose brain is more complicated? Our brain, right? Because we can think, monkey cannot think, right? Yes, we have less than Neanderthals. Yes. So your Homo sapiens have this 13,500. So this is your brain size. So this is D. So we go. Then Homo habilis is the first one. Then we have Homo erectus. Then you have Neanderthalis. Okay. So Homo habilis will be your least. So this is the first one. So you go for the least. So this is A. So Neanderthalis, so the African man who's very similar to the monkey, he has a huge brain size. So that is B is 4. And then Homo erectus is in between. Okay, so please don't get wrong or get confused like how I got. Okay, so this is the right match. So don't worry, for all those who are new, I will teach evolution. So question 12, a gene locus has two alleles, capital A and small a. If the frequency of the dominant allele is 0.4, then what will be the frequency of the homozygous dominant, heterozygous and homozygous recessive individuals in a population? So this is based on your Hardy-Weinberg principle, once again from the chapter evolution. So here, Hardy-Weinberg's equilibrium or equation is p square plus 2 square plus 2pq is equal to 1. So this is nothing but AA small a plus 2 heterozygous is equal to 1. Yes, 2 heterozygous is equal to 1. Okay. So if they say the frequency of the dominant allele, so this is p square. So p is only a. So here P is 0.4, so P minus Q, so P plus Q is 1, right? So 1 minus P is equal to Q, so Q is equal to 0.6. Now they want the frequency of homozygous dominant. So this is homozygous dominant, they want heterozygous and they also want homozygous recessive. So square this, so this is P square. So P square is nothing but AA. We need AA. So this is 0.16. So this is A small a. So this will be 0.36. So AA will be 2PQ. So that is 2 into 0.4 into 0.6. So this will be 0.48. So now give me the answer. So A is 0.16. So AA is 0.48 and A is 36. Yes, option A is the right answer. So this is how you have to solve. This is Hardy-Weinberg equation. Okay, so Shimpi does not know. I will teach you. But yes, questions of this type also comes. So question number 13. Variations caused by mutation as proposed by Hugo D. varies are small and directional. Small and directional is random and directional, random and directionless. So Hugo D. varies proposed saltation, huge large step mutation. So he said they are random and they are directionless, right? Yes, they are random and they are directionless. So this is what Hugo D. varies argued, whereas Charles Darwin said that they are small and they are directional, right? So this is option D. So question number 14. So in a species, the weight of a newborn ranges from 2 to 5 kg. 97% of the newborn with an average weight between 3 to 3.3 kg survives. Okay, so average from 3 to 3.5 only survives. But from 2 to 2.5 or 4.5 to 5 kg, they die. So what is this type of selection? Disruptive, cyclical, directional or stabilizing? So type of selection is not discussed in detail, but since only a particular natural selection is working here, only one group is surviving, so they are very, very stable. So this is known as stabilizing selection. Okay, so this is option D. So with this, we are done with your genetics and evolution. So in the next lecture, I will discuss reproduction and the other 3 chapters, other 3 units. Okay, yes. So I hope you enjoyed this question session. I don't want to saturate you because the human brain cannot concentrate on more questions. So that's fine. We'll stop it for now. So please join me at 6:30 p.m. for the rapid fire Menti, and please be prepared. So please be very quick also. So don't come if you're sleepy and if you're hungry, because you will not be able to concentrate. But just play. It's a game at the end of the day. Alright. So thank you so much for joining my lecture. So please do take care. And this is me wishing you more and more. So looking forward to meet you at 6:30 p.m. as well. So until then, take care and bye.